Okay, well, I'd like to welcome everybody to our second IMAG MSM Working Group meeting of April 1st. Thank you, everybody, for who joined us at 11 a.m. this morning. Today, we will be following our usual meeting structure. That is, we'll have two brief talks and then an opportunity for discussion following the main meeting. So I hope people will be able to stick around to join our speakers for more informal discussion. I need to remind everybody that the meeting is being live streamed on YouTube, and the recordings of the meeting will be made available on YouTube. As usual, the people involved, myself, James Glazier, Indiana University, co-lead with Reinhard Laubenbacher, and helpfully supported by Jim Sluka and Bruce Shapiro, whom you've all communicated with. Anything technical or issues that you'd like to address, please take it up with any of us. We're always available to discuss. As usual, we also have to remind you that we have a Slack channel, we have a Twitter channel. There are the IMAG MSM Wiki pages. There is now an IMAG LinkedIn page. And we have a YouTube channel where all of the previously recorded meetings are available. We'd very much appreciate your help curating and improving the content for these communication channels. They only work if we use them. And curation is time consuming. And we hope people, speakers in particular, are encouraged to share their own talks so that the material that's available is used. Starting last week, we had the opportunity to do some short announcements. 1 minute. If you need to do a slide, that's okay. You don't need to have a slide. And so I quickly call to see if there are any announcements that people would like to make. We had made a comment last week that there were some requests for information on how useful this working group was for people. In particular, if you've made new research contacts, collaborations that this working group has helped you establish, that's an important metric of our success. That could be writing a paper with somebody, writing grants with somebody, preparing resources, or simply extensive discussions and scientific ideas. If you can let us know, especially send that on to Bruce, that's very helpful for us to document the functioning and utility of the working group. It helps us tune the working group. For upcoming meetings, next week we have James Moore and Denise Kirshner speaking. April 15th, Winston Guerrera and Vivek Shenoy. April 22nd, Deborah Peters, and you can see the schedule going forward. We have some people booked already into May. We really welcome your suggestions for future speakers. And as I always say, because these seminars are very, very short, people are welcome to come back and to speak again about material they didn't get to cover in the first time. And we understand that this 15-minute format is a teaser. Sometimes it's frustrating to try to convey all of the interesting work you're doing in 15 minutes. And we're very happy to have people come back and speak again. As rules of the meeting, we'd appreciate it if you're not speaking, if you would mute your microphone so that we can reduce background noise. During the meeting proper, we don't have that much time for questions, so we especially appreciate questions and ideas that are high level and also from the people who don't speak all the time, because there are a few of us who tend to speak a lot. And now we have the breakout rooms for more in-depth discussion afterwards. And I will generally give a 5-minute warning if it looks like people might be overrunning their time so that we can stay on schedule. So please don't be, our speakers, please don't be offended if I simply break in and say 5 minutes. And with that, people know who are regulars that I don't do an elaborate introduction. I don't want to take any more time away from our first speaker. Our first speaker is John Yin from the University of Wisconsin-Madison. He's going to be talking to us about the kinetics of viral growth in cells. So I will let you have the screen share, and please take it away.
Thanks for the opportunity. It's a real pleasure to be here and share some of our interests. So I'll start off with my slide sharing. Hopefully you can see this very busy opening slide. Don't worry, they're not all like this. This is just a little bit of an overview of sort of things we're interested in or have been interested in over the last 20, 30 some years. Quantitative virology in a word. And we've done work with virus counting. This is useful for estimating particle to PFU ratios. We've done a number of studies in single cell kinetics of virus production, showing the high diversity that one can see from both the kinetics as well as the one-step growth behavior. Set up a few real-time systems for dual color gene expression, either by virus host, I'll show an example of that, or by virus and virus mutant. So you can look at the ecological dynamics, either in well-mixed systems or in spatial systems, where green and red here represent a couple of different kinds of virus strains. I actually started off my career doing spatially resolved evolution, showing how viruses growing in a single plaque could expand and evolve. This is a DNA profile of a loss of a gene in a particular kind of environment. And then we've also been very interested in ways that viruses can spread under the presence of fluid flows as well as diffusion. Here are some dishes where there's a little bit of fluid flow going radially outward, and this facilitates the viral spread. And more recently, this is a continuing interest of ours in defective interfering virus-like particles, which seem to be arising ever more frequently in clinical specimens. And these may play a role in activation of innate immunity as well as inhibition of virus growth. And we're also very interested in making viruses for vaccines. And I highlight here Huicheng Shi, who is in the audience. He has done some nice work on Zika virus, very high resolution, multi-samples over multiple cycles. So this is comparing a couple of strains, the African and Asian lineages. So that's a very brief aspect, focusing actually on the measures. And I'll talk a little bit about models then. So we are interested here. I am somehow not progressing on my screen. So let me see if this works. Okay, good. So title of the talk is Kinetics of Virus Growth in Cells. I've got a small lab, 3 graduate students underlined there with a lot of undergraduates, sorry. Graduate students are underlined, undergraduates are other names. Various collaborators at University of Wisconsin and elsewhere. And then I'm grateful for support from NSF and NIH. So we have a number of motivations for studying viruses. Of course, they cause many human diseases, and I should add to that list COVID-19. But there are also other reasons for studying viruses, and that's because they encode the smallest genomes. So if you are interested in how genomes become organisms, you can think of the viral growth as a toy model of development. So virus in a given environment then defines a developmental process. And of course, we have a lot of information about viruses. They've been well characterized not only from genome sequences, but we know something about the way the genes are regulated, how they function, and how they interact. There are short generation times that enable us to do quantitative kinetic experiments on a graduate student friendly timescale. So this is another reason for studying viruses. It's nice to try to get on the same page comparing viruses with humans. So this is a plot of number of proteins encoded by the genome and the genome size, where I plotted on the diagonal here 1 protein per kilobase. Humans fall way off this curve, as do many higher organisms. But if we extend this curve to the lower area using mycoplasma as a point of reference, mycoplasma as the point of reference, and all these smaller entities here are viruses, they pack nicely against this curve, highlighting that they tend to be quite efficient in their coding. A lot of the coding that's viral information is actually encoding proteins. So all viruses have in common that they must make protein. So here are various different flavors of viruses, from double-stranded DNA all the way through double-stranded DNA that go through reverse transcription, and various flavors in between. There's SARS coronavirus. I put some stars here on ones where there are kinetic models of growth available. This needs to still be updated because there have been a few more that have come online since this review paper. So let's look at the intracellular growth. Here's a toy version of how we think about modeling. We and the broader community interested in intracellular kinetics of virus growth. You've got a free virus particle that encounters a cell, and various things happen inside when it releases its genome. The genome gets processed to make messages, proteins, viral capsids, more genomes. These get packaged, and free virus is released. So the process of building a model entails going into the literature, translating the biology into the simplest kinds of equations we can possibly imagine to try to describe each step, from initial binding to a receptor, to entry, to processes such as transcription. This is a sort of generic equation for how a given message might be transcribed based on genome, and its presence will also depend on decay. And the protein that corresponds to that message then also depends on the level of the message, how fast it's translated, whether or not the protein gets assembled into other complexes, and also protein decay. So that gives you a little bit of a flavor of the sorts of modeling. And ultimately, in the bottom here, we're producing free viruses. So you'd like to be able to predict how long it'll take and how much you'll make. In summary, here's a process of taking the molecular data from sequences, mechanisms, and gene functions. These feed into the mathematical models, typically coupled ODEs, sometimes algebraic equations, and we've got initial conditions, typically bound binding of the virus to its receptor. And then what you calculate by integrating forward are the various intracellular components, and ultimately, in the red curve here, the progeny virus, where there's a delay time, that how long you need to wait until you see the progeny, and based on the resources, also some level of the progeny that are coming out of the cells. So this process of some version of this has been going on for quite a while, I'd say over 50 years, where I've highlighted in boxes here some of the models that we contributed to, one for phage T7 in 1997, one for HIV in 99, and then vesicular stomatitis virus in 2006. I think it's fun to look back at the very earliest one, this paper in 1971, with Baracelli as the first author. This was virus genetic theory testing by data processing machines, something we call today computers. So this was on some of the earliest computers that had transistors rather than vacuum tubes. So when they were at Institute for Advanced Study, during the day, they were playing war games. During the evening, Baracelli would get in on this computer and do his virus models. There's a nice story of this in Turing's Cathedral by George Dyson. So we and others have developed these models. This slide focuses on works of our own to examine antiviral strategies, to look at how one might infer mechanisms from transcriptome proteome patterns, and how study genetic interactions, epistasis, these sorts of models. One can begin to simulate mutations and interactions, even explore genome design. And nature versus nurture. This is the nature is the genome of the virus versus the nurture, what's the host cell providing in terms of resources for growth. So these are largely reviewed in this 2018 paper. I'd like to take the remaining time to very quickly go over 3 questions that I think might be fun for this audience. How diverse are single cells when infected by single virus particles? Well, you can infect cells with a recombinant virus that carries a GFP. If you do this at low multiplicity, 0.01, then cells that are lighted up, you can pretty much guarantee are infected by a single virus particle. If you isolate those cells and look at the yields from then on the x-axis you have a broad diversity of yields from 10 to 500 here up to 7,500 or 8,000. So about a 300-fold variability in the yield of from these viruses. This is from 134 cells. If you isolate 8 cells and grow them up from the high yield ones, you find they all grow like the average. So there's no genetics in this, at least. And then if you isolate 7 of the low producers, you find that 5 also grow like the average, but 2 grow low. So from this kind of study, you can see that the genetic variability likely plays out in about 10 to 15%, while the rest seems to be from other factors. What are other factors? They might be actually different places in the cell cycle, where you can see that if we grow up the virus in early S phase, we get up about 1,300 viruses per cell. But if we go to G2M, then we can get almost 9,000 virus particles per cell. So there's a 6-fold difference between low and high here, whereas if it were a purely volumetric difference, then you might expect perhaps a 2-fold difference. So there's something special about G2M. Second question, how might we visualize and measure the dynamics of virus cell interaction? Well, we've wired up cells so that the virus comes in and brings in red. And we've also got a system where the cells can be wired up such that interferon-sensitive genes will light up green. And we have a particular mutant that will bring in a red fluorescent protein. So in essence, if this mutant gets into a cell, you can get both red and green. And if we run that experiment, here is at 3.5 hours post-infection, you're seeing a bunch of blue nuclei. But if I let her rip on the movie, then you can see cells light up various colors of red and green. It's a mixture. You even see a little cell running around here before it apoptoses. So if you sit on these individual cells, you can quantify the level of viral activity from red fluorescent protein and the level of host response. This is an interferon-sensitive gene by its GFP. So when you let this rip, you can look at how each individual cell is lighting up and moving across this. This is a dynamic, then essentially a cartoon version of the movie. And you see the red, blue dot is our population average, but what you also see is a huge amount of heterogeneity, both in where you end up and as well as the path that you take. So that's a huge challenge, I think, for modelers. There is also opportunity in exploiting such variability to also gain insights, and I'd be happy to talk about that in the question section. Final question, how fast does a virus grow? Well, it depends on the virus. So a student went into the literature and looked at 101 virus cell pairs spanning diversity of life. This is bacteria, archaea, and eukarya, and pulled out one-step growth curves. We analyzed the one-step growth curves. And if you look at various parameters pulled out of these, like the infection burst size, the delay time, cell volume, cell doubling time, you can look for correlations. And what comes out is that, well, you can look at cells that might be over 100,000 or so variation in size. There's maybe 100-fold variation in burst size. So there's very little correlation between the cell volume and the amount of virus that they make. However, if you look at the doubling time of the cell, so longer doubling time, slower cells out here, and the delay time of the virus, you get this very nice kind of stronger correlation. This is with 100 and 101 pairs. And this suggests that the question should perhaps not be how fast does a virus grow, but how long does a host cell persist when it's been infected? Same cell infected by different viruses may show very similar sorts of growth dynamics. So that's all I had to say. I'd love to have colleagues visit at Wisconsin or send your best students here. We have some nice grad programs in engineering and biophysics, and I'd be happy to take any questions. Thank you.
Well, thank you very much. That was a delightful talk and an immense amount of material in a very short amount of time.
I tried.
If it's okay, okay, we'll ask one question. We'll have one question now And then I think we'll go through the second talk and then have the questions together, if that's okay.
Sure.
Especially since the talks are in some ways very closely related, I think that may work well. Jacob, did you have an urgent question? Okay, go ahead.
Not so urgent, but I'm seeing that he's showing all sorts of movies to expand whatever he's trying to talk about. My question is related to something we're doing here in the dissemination subgroup. Will a format like a journal that publishes interactive visualizations be helpful for you? Will you publish in something like this?
I guess I would have to think about it, because I have been approached by, I think that some of these journals that emphasize the methodology, you know, where they can videotape you. And I guess the reservations I've had is there is an additional cost for that. So it is more expensive to publish for such journals. There's more overhead. So I guess I would have to think about it, but I think there is value certainly in other ways of conveying what we do and conveying the nature of our results. So I was sad that we couldn't show those sorts of animations, and you know, the paper itself is, you look at it and say, you know, it's much less kind of impactful in that respect. We do go into the quantitative analysis though on both of the colors and how they are relating to each other.
You gave a good answer, and I'm not talking about movies. We're talking about actual interaction, but never mind. Thank you very much. You actually gave the exact answer I was looking for.
Thank you for your question.
Okay, thank you. And we'll move on to our second talk.